The action potential is the primary mechanism of intracellular communication between neurons. Neurons, as you know, are the specialized cells of the nervous system in the human body. And so if we look at a neuron right here, on one end we see these tail-like dendrites coming off. In the middle we see the cell body or the soma. And then coming down here we see the long, lengthy axon, which is where the bulk of the action potential really takes place. And so before we jump into the specifics of that, let's break down the action potential first. And so the action potential is made up of two gradients, one being the electrical and the other being the chemical gradient. And by electrical gradient, we're talking about a gradient between positive and negative charge. And by chemical gradient, we're talking about a concentration differential between one type of chemical on the outside, in the case of an action potential, sodium, and on the inside of the cell, you'll have a higher concentration of potassium. And so it's really this concentration differential between these two ions that makes up for what the action potential actually does. And so if we dive into the localized view of the cell membrane here, we can see that sodium is higher, the concentration of sodium is higher on the outside, concentration of potassium is higher on the inside. And on the cell membrane itself, we have three types of channels. Two of them are passive. You have the sodium leak channel and the potassium leak channel. And these simply allow both types of ions to flow down their chemical gradients. So sodium will flow in, potassium will flow out. And this one is an active transporter. It's your sodium potassium ATPase. ATP, because it's an active transporter, requires an input of energy. And the reason being is because it moves these ions against their chemical gradient. And so it brings potassium, moves it in, takes sodium, moves it out. And this is sort of really to maintain this chemical gradient on both sides, because without the chemical gradient, you really can't have your action potential. And so right here, if we see our resting membrane potential to be at negative 70 millivolts, this means that when there's no action potential, the electrical potential at the membrane is negative 70. And so if we go briefly to this chart here, at negative 70 is where the neuron starts. It's where you're resting. And so that's with nothing going on. Negative 70 is where your electrical potential is going to be at. And so when your action potential starts, you have an efflux of potassium ions out of the cell. And so this leads to a raising of your resting membrane potential from negative 70. And so right here we see potassium's going out of the cell. The effect is gradually making the membrane potential more positive. And by more positive, we mean moving away from the negatives towards zero, then towards the positives. And so potassium moves out until we get to here, which is around negative 50 in neuron cells, where we meet our threshold potential. And when the threshold potential is met, that basically triggers the action potential because we're crossing this threshold, and so the action potential is triggered. This leads to even more potassium flowing out, The membrane potential is getting more and more positive until we peak. And the peak is usually around 35 millivolts or so. And at which point your potassium channels are going to close, and so no more potassium is going to flow out, and your sodium channels are going to open, and so sodium is going to start flowing in. And the effect of that is that we're going to start to go negative again. So we're going to start to, and I forgot to mention that this phase here is the depolarization phase. And we're depolarizing because we're becoming more positive. And so the electrical differential between the outside and the inside is becoming decreased because the resting membrane potential is becoming more positive, outside is already positive, and so we are depolarizing. And so if this is the depolarization phase, then the phase after this is going to be called the repolarization phase. Repolarization. And so we're going to keep repolarizing. Sodium's going to flow in, and this is going to cause the membrane potential to become more and more negative. So we're going to repolarize, and we're not going to stop at the resting membrane potential. We're actually going to overshoot it. Going to overshoot it a little bit, and maybe not that drastic, but we're going to overshoot it. And this is actually not accidental. It's actually very important that we overshoot it. This is called the hyperpolarization phase, because we're becoming hyperpolarized. And this is actually really important because if you think about it, if we were to repolarize back to the resting membrane potential, the neuron would be primed right away for another action potential. And we actually don't want that. And the reason why is because we want the action potential to propagate all the way down the axon, okay? We want it to move forward. We don't want it to move backward. And so if an area of the axon was immediately ready for another action potential right after an action potential crossed, this could theoretically allow it to go backwards, and that's very bad. We don't want axons to do that. We want action potentials to only move in one direction so we can have a steady flow of communication. So that's why this hyperpolarization phase is so important. So after hyperpolarization, we have a brief period of latency before we repolarize back to a resting membrane potential. And after which case, we're ready for another action potential.